We're discussing your recent study looking at CD14 positive monocytes in rheumatoid arthritis. Describe for us what this study was designed to investigate.
So basically, we isolated this monocyte from the blood of patients with rheumatoid arthritis, from individuals at risk of developing rheumatoid arthritis, but we also compared this to healthy controls just to check that they were still higher than the healthy controls. And there was three aims. The first aim was to utilize these monocytes to see could we identify a signature of cellular dysfunction in established ORA. The second was to see, well, if this identified signature is a signature of established ORA, is it already present also in these individuals at risk? And the third was that there's this really huge body of evidence and, really exciting field in immunology of metabolism. And we now know that immune cells, when they become activated in these diseases, they change the way they use energy. So once they become activated, they need to it's a bit like a kid in the sweet shop. They need to get energy really, really quickly. And when they get that energy really, really quickly, they become bad. So that's how the immune cells work with regard to metabolism. So they change the way they use energy to help them remain activated and be bad in this environment. So the third aim was really to figure out, so these monocytes, is the metabolism bad, and can we reverse that effect?
How was the study conducted and what was involved?
So as Ursula has previously mentioned, in this study we collected blood from healthy control donors and also ORA patients. So all of the ORA patients involved in this study were recruited from the rheumatology departments at St. Vincent's University Hospital under fully informed consent, and these patients had active established ORA in accordance with the ACR classification criteria. And again, as Ursula has mentioned, we collected these two cohorts, but also we collected another third cohort of this unique individuals at risk who develop aches and pains, but to date have no clinical signs or symptoms of ORA. So once this blood was collected, peripheral blood mononuclear cells, or PBMCs, were isolated. These were then magnetically labelled and sorted to yield a pure population of CD14 positive monocytes. So these monocytes would have been circulating in these individuals and patients' bloods hours previously. So once we isolated this pure population of monocytes, we then ran a number of experiments looking at them ex vivo, so unstimulated, or else under the influence of stimulation with LPS.
What were your findings?
So getting into, I suppose, the findings of our study. So firstly, I suppose, to elucidate the role of these peripheral blood monocytes in ORA, we firstly noted that there is a significant increase in the number of circulating monocytes in ORA compared to healthy control. And then we ran an inflammatory gene array, and really what we need to kind of focus on here are these red bars, which indicate those inflammatory markers which were hyper responsive in our ORA monocytes compared to healthy control. And as you can see here, that's quite a lot. And just to, I suppose, pick out a few of these genes, we further validated three of them here, and you can see that these are significantly increased in our ORA activated monocytes compared to healthy control. And actually what was interesting with the genes that we found was that this inflammatory signature was very reminiscent of what an inflammatory M1 macrophage would produce in the joint. So those cells that the monocytes can become when they get into the joint, the dendritic cell, the monocyte, the osteoclast. So we think that these cells are perhaps primed or poised to become inflammatory macrophages once they leave the blood and enter the joint. So again, as Ursula kind of mentioned earlier about the kid in the sweet shop type thing. So these cells, we know that a kind of a greater inflammatory burden, which we've just shown that these cells are hyper-inflammatory, is often associated with a greater burden or greater demand for energy, or ATP. So firstly, looking here at the kind of mitochondria. So we know that the mitochondria are the powerhouse of the cell. And really here, what we can note is that we get a mixture of these regular and irregular shaped mitochondria in our ORA monocytes compared to healthy control. But then just to, I suppose, further elucidate the role or the metabolic competence of these cells, we use this machine, which is called a seahorse flux analyzer. So the seahorse machine, all it does is it measures the two major bioenergetic pathways of the cell in real time. So you've got glycolysis and oxidative phosphorylation. And you kind of get graphs out looking like this, where you can get a reading of how much the cell depends on one pathway versus another, or do they depend on both. So what we found here was that these cells have, a striking increase in their glycolytic capacity. So in this metabolic pathway called glycolysis, which is known to be associated with inflammatory function of cells. So glycolysis is this pathway whereby it's quick, you get a lot of ATP. Immune cells love it because when they want to get, activated quickly, they don't want to go through the whole mitochondrial respiration, oxidative phosphorylation. It takes too long. But the problem with glycolysis is it's quick, but they kind of burn out quickly. So they don't get a huge amount of energy at the end in comparison to mitochondrial respiration. But really here, what we're seeing here is this boost in glycolytic capacity of our cells shown by these traces. And again, this is further paralleled by significant increases in some key glycolytic enzymes such as PFKFP3 and one of the master regulators of cellular bioenergetics, which is HIF1 alpha. And just looking here then, I suppose, at the metabolic phenotype profile, you can really see this switch from an aerobic, quiescent phenotype towards this hyper-energetic, hyper-metabolic phenotype in our ORA-activated monocytes. So we do see that both pathways are boosted. However, when we look at our ECAR to OCR ratio, we can see that it's actually in favor of glycolysis. So even though both pathways are up in these cells appear to depend more on glycolytic mechanisms than oxidative phosphorylation. And actually, when we inhibit glycolysis using 2DG, we see a complete abrogation of our inflammatory signature with this inhibition of this metabolic pathway. And not shown here, but when we inhibit the other metabolic pathway, we don't see this decrease or resolution of inflammation. So we were also interested in other regulators of this phenomenon. So STAT3 we know is implicated as a transcription factor. It's implicated in the invasive mechanisms of ORA. There's also been links between STAT3 and metabolism. So we thought this would tie in nicely. And when we looked for it, luckily STAT3 was significantly significantly enriched in our ORA monocytes compared to healthy control. And actually, when we block STAT3 using static, a small molecule inhibitor of STAT3, again, we see this resolution of inflammation. So we get a complete decrease of our inflammatory signature in the presence of this inhibitor. And actually, again, we get a decrease in the metabolic capacity of these cells. So this hypermetabolic boost that these cells were under, we can kind of return that to baseline in the presence of a STAT3 inhibitor.